Assalamualaikum warahmatullahi wabarakatuh and hello. Let's learn about steps in gene cloning. The first steps in gene cloning is isolation. What happened during isolation? Plasmid is isolated from the bacteria. The example of bacteria used in here is E. coli. Let us look at the structure of plasmid first. Plasmid has selectable genetic markers. For example, is ampicillin resistant gene and also laxi gene. Within the laxi gene consists of restriction site. Restriction site is a site where the restriction enzyme will cut. Alright, now foreign DNA also will be isolated from the donor cell. This foreign DNA may contain the gene of interest. So this is the example of the foreign DNA, the green color. So we already finished for the first step, which is isolation. The second steps in gene cloning is cut or cleave. Plasmid is cut open at restriction site within the laxi gene. For example, we use the enzyme EcoR1 and it will produce the open plasmid. For the foreign DNA is also cut using same restriction enzyme, which is the EcoR1. This restriction enzyme will produce sticky end or blunt end, but in this case we use EcoR1, so it will produce sticky end. So both of the plasmid and foreign DNA will have complementary sticky end. So we already finished for the second step, cut or cleave. Now we go to the next step, which is the third step, which is insertion. During insertion, the DNA fragment containing the gene of interest will be inserted into the open plasmid. And then DNA ligase will catalyze the joining between this DNA fragment, the green color, and the plasmid by formation of phosphodiester bond. Then it will produce recombinant DNA or recombinant plasmid. Finish for the third step, which is insertion. The next step is transformation and amplification. So what happened to this recombinant DNA? This recombinant DNA will be reintroduced into the bacterial host cell by transformation. Once the bacteria accept the recombinant DNA, we can call the bacteria as recombinant bacteria or transformed bacteria. Then this transformed bacteria will be amplified. So the recombinant DNA also will be amplified. How? The bacteria will undergo amplification and form a colony. How this bacteria can form a colony? Okay, these bacteria are plated on medium. For example, in this petri dish consists of a medium of ampicillin and also agar. Ampicillin is the example of antibiotic. Bacteria with plasmid have ampicillin resistant gene. So look at this yellow color of plasmid. The yellow part is the ampicillin resistant gene. Ampicillin resistant gene meaning that this gene is resistant towards ampicillin. So bacteria with plasmid with the ampicillin-resistant gene will be resistant towards ampicillin. And this bacteria can reproduce to form a colony. So by looking at this in this petri dish, either it is white colonies or blue colonies, these bacteria have plasmid with the ampicillin-resistant gene. Bacteria without plasmid cannot survive because the function of ampicillin here is to kill the bacteria without plasmid. The last step is screening. Bacterial colonies containing the recombinant DNA are identified using blue-white screening. Now, let us look at the white colonies first. The white colonies shows the bacteria contain recombinant DNA. Let us look at the structure of recombinant DNA. So this plasmid, which consists of the lexi gene, is already disrupted by the DNA fragment of foreign DNA. So the lexi gene is already disrupted, so it cannot function. And as a result, the beta-galactosidase cannot be synthesized or produced. When there is no beta-galactosidase, so it cannot hydrolyze the X-gal within this medium. So the colonies will form white. Next, we go to the blue colonies. The blue colonies shows that the bacteria contain non-recombinant DNA or non-recombinant plasmid. Now, let us look at the structure of non-recombinant DNA or non-recombinant plasmid. So this plasmid has intact lexi gene. So the lexi gene in here is function. So the beta-galactosidase can be synthesized or produced. And when this beta-galactosidase is present, so this beta-galactosidase can hydrolyze the X-gal and it will turn to blue color. From these white colonies and blue colonies, we need the white colonies because these white colonies contain recombinant DNA, which consists of the gene of interest. And as a conclusion, the last point, the recombinant DNA is amplified. Okay, so I think that's all for these steps in gene cloning. Thank you.